Hello, everyone. Thank you for joining today's DoD Environmental Planning and Conservation webinar series presentation. Today's session is titled A Range-Wide Western Pond Turtle Status Assessment on Military Lands, Occupancy, Population Structure, and Pathogens. Today's presenters are Tom Akre, Conservation Ecologist, Smithsonian National Zoo and Conservation Biology Institute, Emily Asche and Matt Parry, both graduate students at the Illinois Natural History Survey, Curry Research Institute, and Natural Resources and Environmental Sciences at the University of Illinois, Urbana-Champaign, Rob Lovich, Naval Facilities Engineering Command Southwest, Matt Allender, Director, Wildlife Epidemiology Lab, Department of Veterinary Clinical Medicine at the University of Illinois, Urbana-Champaign, and Mike Dreslik, Director of the Population and Community Ecology Lab, Illinois Natural History Survey, Curry Research Institute, University of Illinois, Urbana-Champaign. Today's presentation will be recorded and then posted on the Natural Resources DNX website under Resources. Before we get started, please take a moment to make sure your phones and web browsers are on mute. With that, I will turn it over to Tom.
Good afternoon, and thanks so much, everybody, for joining us today. I'm excited to be here, presenting to you all on behalf of our team, the first part of our range-wide Western Pond Turtle Status Assessment on military lands, focusing on detection and occupancy of turtles across the distribution and bases, their population structure, and their pathogens. And let me just take a moment to tell you who we are. So as we said before, I'm a conservation ecologist at the Smithsonian National Zoo and Conservation Biology Institute. Speaking in introduction on behalf of Mike Dreslik and his team, Emily Asche and Matt Parry at the University of Illinois, who conducted the majority of the field work and analysis on this project. Matt Allender at the University of Illinois Wildlife Epidemiology Lab, who did all of the work on the pathogens. So we're excited to talk to you today about this project that's 2 years in the making at this point, that came together through the Chesapeake Ecosystem Studies Cooperative, specifically the Chesapeake Watershed version of that cooperative with Department of Defense and the Smithsonian Institution. So thanks so much to the Department of Defense Legacy Resource Management Program for funding this project and being supportive of these kinds of projects that focus on understanding the status, the distribution, the abundance, and the threats to these DoD mission-sensitive species, such as the Western pond turtle. So let me give you a little bit of background on the Western pond turtle. I know many of you who are resource managers in the West know a lot about the Western pond turtle, as it is on your installations or in other places where you're focusing your research and management. But for the rest of you, the Western pond turtle, Actinemes marmorata, the northwestern pond turtle in blue in the range map on the left, and Actinemes pallida in brown in the range map on the left, are currently considered vulnerable on the red list assessment of endangered species. But that probably will change to endangered beginning next week when the specialist group comes together and does a lot of North American assessments. And that's in part because over the last 2 decades, a lot of research has been done across the range, particularly in California, that demonstrates that the Western pond turtle populations are in decline. Both species populations are in decline across a lot of their range. From the kind of standard usual suspects of things that are affecting turtle populations across North America and across the world. And that's primarily habitat loss and conversion, land cover change, and climate change. In this case, for Western pond turtles, the effects of drought on these populations, and then either invasive competitors or invasive predators, in this case with Western pond turtles, red-eared sliders as competitors, and bullfrogs as predators of early life stages. And that's because Western pond turtles rely on these wet habitats, these sloughs, these rivers, these creeks, these ponds across their distribution, oftentimes in fairly xeric habitats where drought can have profound effects on their overall life history. And so as a result of all of these different threats in singularly and cumulatively, these populations are in decline. And so there has been a need for quite some time to kind of come together and understand the status of these species across their range and develop capacity for conservation research and conservation management. So we stepped into this framework, that is, this team stepped into this framework behind the existing regional conservation cooperative, of which Rob Lovich and others were part, which has this mission to develop a comprehensive and coordinated group of stakeholders in order to manage, conserve, and research the species in perpetuity through keeping long-term viable populations of wild western pond turtles. Across their range, there have been several different listing categories for western pond turtles, including the current evaluation by the U.S. Fish and Wildlife Service, that is the listing consideration under the Endangered Species Act. But in Washington, the northwestern pond turtle was considered endangered because of declines. In Oregon, it's considered a sensitive species. In California, it's considered a species, or those species are considered special concern. And overall, as is the focus today, it's considered a DoD mission sensitive species. As I said, there was also a species status assessment, which was completed last year, attempting to understand the distribution and abundance of the northwestern pond turtle and the southwestern pond turtle across the range as it relates to a series of factors or threats which are responsible in either directly or indirectly for declines in populations. And so an output of that was these prediction extinction models for western pond turtles, the northern one and the southwestern one, as it relates to land conversion, climate change in the case of drought, and bullfrog presence, essentially trying to understand the likelihood of extinction of these species and whether or not they warrant listing. And this is particularly important as it relates to our discussion today and our work over the last couple of years, because the Department of Defense manages so much land where the western pond turtle, particularly in California, is historically known from and currently potentially going to exist. Indeed, DoD does manage a lot of land for threatened and endangered species and ecosystems, a disproportionate amount because of the size of their lands and the distribution of their lands around the country. Around the country where endangered species are clustered in California and in Florida, for example, and around the country in the Southeast and the West, where endangered species and a high diversity of species and high diversity of endemic species are clustered. And so we see, as the case with amphibians and reptiles, the western pond turtle is very well distributed on DoD lands in California, and therefore groups like Department of Defense Partners in Amphibian and Reptile Conservation, DoD Park, led by Chris and Rob, are important components of the conservation of the western pond turtle in order to build multi-agency partnerships such as the RCC for understanding how the DoD can balance mission readiness and partnering with other federal agencies, state agencies, and regional agencies to share goals and strategies for effective conservation programs that allow for resilient and adaptive approaches to balancing the needs of DoD lands or other federal lands or state lands with preserving species. And so one output, which is critical to the establishment of this project that Chris and Rob and others undertook, was understanding the distribution of the western pond turtle on military installations, in this case in California, where they found in 2018 that it occurred on 12 or 13 installations across all divisions and potentially occurs on several more outside of California, in Oregon and Washington. So as part of this cooperative agreement through the Cooperative Ecosystem Studies Unit, the Chesapeake Watershed 1, Smithsonian and DoD undertook this project, that is a range-wide western pond turtle status assessment, in order to first maintain a comprehensive inventory of western pond turtle populations across their range, gather updated biological information in order to support effective management, to educate and train personnel on the installations engaged in western pond turtle management and conservation, to strengthen partnerships around the region and on and beyond installations in the military in order to foster conservation efforts and develop a framework for continuous monitoring and data sharing and understanding trends and patterns for these turtles on military lands and beyonds. So like I said, we came together with the University of Illinois, in particular, Mike Dreslik's lab at the Illinois Natural History Survey and Matt Allender's lab at the Wildlife Epidemiology Lab through DoD Park with support of California Department of Fish and Wildlife to understand the status and distribution and abundance and population structure of Western pond turtles in California. And we were fortunate that we were standing on the shoulders of lots of really capable people who had done a lot of this work for a long time. So we had monitoring protocol standards for visual encounter surveys and trap surveys and all of the important techniques for sampling the turtles and understanding detection, occupancy, abundance, and population structure to stand on and move forward. And so I'm going to tell you a little bit about our first objective, and then the rest of the team is going to tell you about the rest of the objectives. So one of the things we did over the last year and kind of culminated in a report that Rob Lovich gave to the RCC this time last year was to collate historical data on Western pond turtles on the 13 military installations. And our goal was to compile a comprehensive database for these installations so that we understood what kinds of records were being kept, what the extent of those records, what the gaps were, what kind of protocols they were using for collecting and recording that information, and whether or not we could train them further to equip them, that is the natural resource managers, with enhanced field monitoring skills in order to develop this kind of long-term benefit of developing a standard data resource and field protocol resource for future Western pond turtle studies. We thought that developing a centralized repository for Western pond turtles would allow for continuing ongoing monitoring and analysis. And so that was one of the goals that we think is an important feature of what we've done. We also thought that developing a field manual ensured uniform data collection across sites, both within and beyond installations, which would allow for understanding finer grain trends within and beyond sites, and therefore allow for kind of more better informed status assessments. We thought that training the teams on these installations would increase capacity for updated information moving forward. And we thought that developing this database would allow targeted conservation in reflection to this trained team and these protocols. And all told, we thought this would be a foundation for future monitoring and research. So the team visited all 13 sites in 23 and 24. All sites were visited in each year, and some sites were visited twice in 1 year, all the way from Camp Pendleton and Fallbrook down in the San Diego area to Beale and Travis north of Sacramento, all across the large scale landscape of California along the coast. Along the way, in order to develop this database and create a training framework, we had initial meetings with resource managers to understand the data quality and depth, and then one-on-one meetings in the field or group Zoom meetings in order to understand the limitations of the data, in order to develop an insight into the current data and guide the database structure with the idea of supporting the development of this tailored monitoring strategy. Obviously, we wanted to create this database by looking at what they had done and tailoring what they would move forward in doing with what we were doing in order to understand detection and occupancy and abundance and population structure, so that we could essentially create a framework for military and research partners to actually take on this monitoring strategy. So if we look at Fort Hunter Liggett as an example, south of San Francisco, we can see that, beginning in 2021, they started monitoring Western pond turtles on site. And in 2023, we joined them. So they had 3 years of data as of last year when we started to put together this compilation of captures and recaptures and distribution of information for the Western pond turtle on this site. But we did this for all of the installations, like I said, all the way from Warner Springs, south of San Diego, to Beale up in the north, so that we had a cumulative set of information for each installation going all the way back to 1993 in some cases, that allowed us to understand the total number of captures and the average number of captures per base across the range. The database is actually more comprehensive. This is just a snapshot. And then ultimately, we wanted to develop training resources for staff that allowed us to understand all, and them to understand all of the features associated with developing a protocol for training. And with that, I'll turn it over to Matthew Parry, who's going to talk about Objective 2.
Well, hopefully everyone can hear me okay. My name is Matt Parry, and I'll be speaking about how to determine detection at the 13 installations that we surveyed. So the goal is to establish a detection probability tool for the Western pond turtle. We did this by conducting trapping sessions across these bases to assess presence. And then we want to also look at environmental variables to optimize these survey results and efficiency. Because the future application of this will be for cost effectiveness, because we are well aware that many of your resource managers, you guys have plenty on your table. You have a lot of different T&E species you have to work with and other environmental projects. You're good, Tom. So how do we did this? How did we obtain our data? So with our trapping design, we used, as you can see on our right, we use both hoop and minnow traps to meet different areas of water bodies, deeper waters as well as shallow. And we also recorded GPS, microhabitat, and turtle metrics, so morphometrics, as well as giving each individual turtle their own mark, which Emily will get on to later. Our survey season lasted typically from the end of March to the end of June. We did go to July on our second season. And then additionally, we also grabbed disease swabs, blood samples, and photographs of each individual turtle. So to help with this detection model, we also need to look at weather data and other covariates to help us assess our model and to give it more strength. And so there's a ton of options we can look at, landscape data, habitat data, weather data, microhabitat data, and et cetera. And so I really wanted to focus on variables that resource managers or whoever is deciding to go out there and trap for these turtles would be able to easily do and not necessarily have to spend a fortune on equipment to be able to assess this. So like many low density and secretive species, they're hard to detect. And so you could be putting a lot of hours in just trying to even get one in some places. And so we want to look for optimal conditions. And so that analysis will help us identify favorable survey conditions to where someone can go out and trap and have the highest ability to be able to detect a Western pond turtle. You can go next one, Tom. So I decided to look at many variables, as you can see on the right. At the top, you'll see the DACTN. So that is the response variable, so that is the presence or absence of the Western pond turtle if we came up to a trap and there was one in there, if there wasn't. And then I added in trap day, site, substrate, average canopy cover, and then also a lot of temperature related variables. So mean max mins for previous days and 3-day lags and 7-day lags. And so with all these variables, you're going to look to the right and you're going to see this correlation matrix, and you're going to see a lot of dark towards the top. And you're going to see that it's all relating to most of the temperatures, because obviously they're going to be kind of explaining themselves and it's going to become very redundant with the temperatures. And so if you look to the left, I did a VIF table, and the primary purpose of the VIF is to measure how the variance of the regression coefficient is inflated due to multicollinearity. So as you keep going down the table, you're going to see the VIF just really increasing high. And we get all the way to the bottom of the 3-day mean, it's at 1,000. That is massive offender. And so we can't have that. It's going to really reduce kind of the stability of our models. And so I decided to shrink it down to looking at just minimum temperatures, because those were the least offensive when looking at the parameters in our fixed effects that we'd be using. So these are all the candidate models that I decided to look at. And so at the top, you're going to see your global min. And so this is including every single variable that we just looked at to that condensed size. So all the minimum temperatures, average canopy, and then global horizon, horizontal irradiance. And then the next one will be the null. So this will be none of those fixed effects. This is purely just going to be the response and then the random intercept that is being used to, whether the detection is happening or not. And then a few other, you're going to see that I use site for many of them because I truly believe that site was a huge factor with detection. Many of the sites produce either very extremely high capture rates or low. And so I wanted to see if site was going to influence a lot of what we see when comparing these models together. And so it's good and it's bad. So the global min ended up being our top model. It performed the best. You can see that the change in AIC has the lowest ranking at the cost of, there are many parameters being used. There's 33, and that's going to induce a penalty for each of those parameters to make this fit. So we don't want to have any overfitting in these models. And you also see that at the top of there's a WI, so that's the weight, and it produces the most weight. So that is the evidence of each model, basically telling that this is the best among the set. Another thing I wanted to note is the R-square values. So there's marginal and conditional R-square values, and this is basically explaining. So the marginal R-squared is basically the statistic that quantifies the proportion of the variance in the dependent variable with the fixed effects. So these fixed effects are what's kind of towards the bottom. You're going to see previous day min, day of min. So these are all those covariates that we looked at, and we're going to see how those are performing. And so you're going to see with the marginal, it's at about a 0.29. So that's what's explaining the variance with the models. But then we look at the conditional, this is also including the random intercept. And so I stated that earlier, and that is discussing this unique identifier that I added into this code to basically give each trap night. So each individual trap, each day that we checked it, has its own unique code. And so what it's kind of telling us there is that is also giving us it's also very it's an additional, it's very important that we have that intercept in there to explain what is happening. And another nice thing to see is that the null is at the bottom. That's saying that, so these fixed effects are having some sort of influence on detection. It's not just, these fixed effects do matter, even though, as you can see with their R-square values, they are very low, but they do contribute to detection. So with these models, you have to test them. You can look at the AIC table and you're going to say, okay, yeah, it says it's top, but how strong is this model? And so I did some response operator curves for our top two models that we had, and that was the global min and trap location. And as you can see, they both have a strong predictive capacity. So if perhaps say if this the line was following that 45-degree angle that's falling up, that would basically mean that this is happening by random chance. And so this is basically telling us that the AUC of 0.93 is suggesting that both models have a very high discriminative ability to kind of distinguish between positive and negative effects between the detection of turtles. So let's flesh this out a little bit, just because we can have variables, covariates. So let's look at someone that we've been focusing on. So as you can see with these density plots, the whole point of these is to kind of create a smooth visual representation of how these variables in the data set are distributed across the range. And so what you're going to see immediately, and I can see this just by looking at it right now, the minimum temperatures, they all kind of have a very similar peak. They all have kind of a peak between 10 and 15 degrees Celsius. You look at average trap cover, it's really high towards zero. Many of the sites that we went to, they didn't have a lot of trees. You go to Beale and there's not a tree in sight at some of those at the lakes. And then also with global horizontal radiance, this is an average. So this is basically the sun that is meeting the horizontal plane. So if you think about probably some people who have seen Western pond turtles, you're going to see them out basking. They're enjoying thermoregulation. And so that was another factor I decided to look at. So let's look at the meat of all this. So these are the effects of all the covariates that we looked at with detection of Western pond turtles. And you're going to see there's a lot of uncertainty with many of these. And if we look back at the density plots, you're going to see that due to this uncertainty, it's just because we didn't capture this moment, you know. If you look down at the mean 3-day minimum temperature on the bottom left, you're going to see our confidence intervals just go crazy after 15 degrees Celsius. And that's just because we didn't capture that temperature past then. And so this kind of comes down to, you know, if we are able to have a long-term study where we have trapping, you know, in each month, because we were only there for the 2 months during the most optimal time, this could definitely straighten out many of these effects. And another one I really wanted to note was trap night. So that's in the bottom middle. And as you can see from the first trap night, we're hitting a steady decline as we keep increasing our trap nights of each trap that's out. So is that due to trap shyness, the turtle's getting too smart, or what? But it definitely brings in back to, as a resource manager, you don't necessarily have to keep these traps out forever. You know, you want to have them kind of in a more of a rapid succession to have the most success, while you're also not creating trap shyness and losing that ability to capture these turtles. So I stated earlier that sight was a huge contributor to detection, and I really do want to emphasize that yes, it is impactful towards detecting a western pond turtle, but it doesn't necessarily mean we're going to catch like millions compared to none. So I really wanted to emphasize that. But if you just take a look, many of the sites, the detection is very low. It can be challenging, but there's also a lot of variance between all of them. And you look at Warner Springs and you see that massive error bar, and that's just because we had the lowest time of there. We were only there for 3 trap nights compared to most other sites that had more than 4. So there's going to be a lot wider margin there. But then also with the substrate, you look and you see that packed mud, sand kind of has a higher ability of detection compared to cobble or rocks or even silt.
All right.
Okay.
Well, with that,
Hi, everybody. Sorry, Tom. I can introduce myself. Hello, everybody. I'm Emily Asche. I'll be going over Objective 3 with you guys, which covers abundance, population structure, and demography. Okay, to kick it off, there were a couple different goals within this objective, which included assessing the western pond turtle, abundance and population demographics across the 13 bases that we visited. Data collection to achieve those goals included recording different body sizes, stage, and sex. The analysis methods for these goals varied a bit, especially in complexity, and some of them were very dependent on the quality and the quantity of the data that we were able to collect. Another major goal of this objective, as Tom mentioned previously, was to train the staff on our trapping and workup protocol so that hopefully upon the end of our project, the installations can continue on our monitoring efforts, as that is what's going to really boost the demographic insights that we collected. The more years that you're spent monitoring, the better the insight. Okay, the expected outcome and insight that we hope to get from our monitoring include population robustness, any demographic red flags, or population vulnerabilities. Also, of course, to set that foundation for monitoring for the installations in future years, which could then lead management efforts and guide where they need to be focusing on. Okay, I want to cover the difference between our trapping methods between the 2 years briefly, as that is seen in our results later on. So in 2023, we had 50 aquatic traps left out all week. So they were set Monday and pulled on Friday. Thank you. In 2024, we had half the amount of traps and left out half the amount of time. So we set 25 aquatic traps on Monday and pulled on Wednesday and set in a new location from Wednesday to Friday. Okay, getting into one of our goals, which was estimating abundance. It was a bit of a tiered analysis, as I mentioned before, highly dependent on the quality and quantity of our data. So our options were a CPU-E for low capture and mixture model for count data, and CMR for recapture data. We had to be a bit flexible with our models depending on the data, and we were able to get CPU-E values as well as end mixture model for the estimated abundance. CMR, we didn't get to that. It is much better suited to long-term studies that get a lot of recapture data. So we did a couple of those alternative models with the CPUE and the N mixtures with the goal in mind to get the most reliable abundance estimate for each base. Okay, getting into those estimates a bit. These are some results that Matt Parry put together for me. This is a CPUE table, a catch per unit effort. Those values on the right side are what we're using for that estimate of abundance. The value is gathered by dividing the total capture by the hours spent trapping. So you can assume that the lower the number, the lower your abundance, as it took more effort to catch few turtles. The higher the number, you can assume there's a higher abundance. If the bases continue on monitoring the way we instructed them, they can look at those values across successions of years to see if that abundance estimate is going up or down. Okay, here is the raw count data I wanted you to see briefly before I go further into my estimated abundance and mixture. So in 2023, we got 261 unique individuals. In 2024, that dropped to 119. That again could be, as I mentioned previously, our trapping method between the 2 years. We had fewer traps for fewer nights in 2024. The table there is the raw count data. The X's represent years that we did not visit that installation. I want you to note how variable the total count data is for the bases. So going further into my analysis, I did select the installations that had sufficient data to base the results off of. You can continue, Tom, thank you. Here is the results for my N mixture model. You can see that we broke it down by specific installation, further into the unique water bodies sampled at the installation. Some of those installations had several different unique water bodies. And then down further by year sampled, and then the EST column are the estimated numbers in that unique water body. They're very variable. The lower numbers, those can reflect potentially not as viable populations. For example, the Nacimiento River at Camp Roberts, they have very few estimated abundance. However, you have to take that with a grain of salt, as Bass Pond is somewhat connected to the Nacimiento, so they're just not inhabiting the river as frequent as the pond potentially. Here is just a different way to visualize those estimated abundance. These are the results from that N mixture model. If installations continue on our monitoring efforts, you'll be able to plot that and potentially see a trend. So is the trend of the estimated abundance going up over the years? Is it going down? Is there cause for concern in your population? Okay, moving on to the next goal, which was population structure. With this goal, we gathered sex ratios, stage ratios, size frequency distribution, and allometric growth rate plots, as well as a little bit of reproduction. The goal from all of these analyses was to identify any patterns in the demographic structure that could indicate weakness or vulnerability. Here are some graphs I put together to visualize the sex ratios and the stage ratios. Again, these bases you see are just the ones that had sufficient data to do the analyses on, so it doesn't include all of them. But you can see that males typically outweigh the amount of females caught, and in stage ratios, in every instance, adult heavily outweighs juveniles, which is not necessarily cause for concern, but if you monitor those trends, you can see, is there an alarming pattern in that you need to understand why those ratios are so skewed. Here are some tables from those ratios done with a simple binomial exact test. The p-values are indicating whether the bias in these ratios is significant. So is there a significant amount of males versus females and adults versus juveniles? In the sex ratio instance, there were some significant values, none of them entirely alarming. However, you should monitor those and understand it further again to understand, say, why there are fewer females on your installation, as that could lead to low recruitment, such that could then lead to a population crash. The table on the bottom are some site-specific p-values. Sex ratios, they weren't too bad. Again, stage ratios, all very significant. Not to be alarmed by those, but again, to be further understood. Here are some histograms showing size frequencies binned by adult and juvenile and female and male. You can see it's kind of all over the place. That really reflects how many different populations we surveyed, as well as how we work with both southwestern and northwestern pond turtle, which are known to have some size dysmorphia. This is great to look at to see if you have any missing size classes, which could indicate the health of your population. So if you saw a missing size class in one of these bins, say you are missing subadults, that could indicate that in 10 years or so, your population may be crashing as those subadults are now adults and there are fewer to put back into the population with recruitment. This is just the plastron measurement. The first one was carapace, just different way to visualize the different measurements, can tell you the same thing. Here's some allometric growth rate plots. These are male specific for the different morphometric measurements we took. You can, this just gives you an idea of the growth rates in relation to each measurement we took. So you can see that shell height on males is not necessarily growing as fast as, say, carapace width or plastron length. Here are the female allometric growth rates. You can compare male and female. You can see that the female shell height growth rates are higher in comparison to males, along with the carapace width. That is, you can chalk that up to they need to have larger body cavities to fit a larger clutch size. Okay, getting into reproduction, our last thing a little bit. This is tough. Just to explain a little bit, females are known to reproduce in alternating years in some populations. They can have anywhere from 1 to 13 eggs. So when we palpated the female's body cavities for eggs each year, maybe it was an alternating year and they weren't reproducing in that specific season, or maybe they had so few eggs we couldn't feel it in the body cavity, but the overall percentage of gravid females that we came up with was rather low at 32%. Most of those installations that you can see in the table, I included month too, just as reference. They mate and nest through March to June. They're relatively low. I will point out Point Magoo, which is the highest at 85%. We were there, we found them in the channel where we assume they're staging to nest. We think they nest in the residential area that they were behind. So maybe we caught them all at a great time, and if we'd been there in the next few weeks, it would have shown that none of the females were reproducing. So maybe it was a timing issue, but it's definitely something that installations should look into in future monitoring to really understand the timing and where females are nesting and if this is cause for concern. Most of my results, they're reliant on, it would be great to get extra monitoring efforts in to further understand the results that we're seeing and build on the analyses that we've done so far. So that wraps it up for my objective.
Thanks to Emily and Matt, and on to Matt Allender for disease surveillance and monitoring.
Hey everybody, how's it going? All right, so the objective of this one was to identify presence of pathogens, in particular Emidomyces, that is causing major issues with the northwestern pond turtle conservation efforts, especially in Washington State. This is a major concern in that it is fatal and transmissible. So the Emidomyces, just really quickly on this, is that it's a fungi just in a class very similar to Ophidiomyces and snake fungal disease. It will eat away through keratin, has a large host diversity. We've had over 40 species of turtles worldwide that have been diagnosed or detected with Emidomyces. There's lots of unknowns. We are in the early stages of understanding this disease, and the studying of it has been difficult because it goes into the bone, and that's very difficult to detect in an antemortem sample. What you're seeing here are pictures of a Blanding's turtle, and then now here is northwestern pond turtles, and you can see these lesions on the external surface, and when you radiograph them, you can see the holes in the bone. Anything black is a hole, and so the internal cavity of the turtle or tortoise or whatever is affected is completely open or maybe covered by a little bit of keratin, but in general, this is what eventually leads to death. But this is a slow process. This takes years to get to this point. And the conservation concern is at this point, you can't release positive individuals, and it seems that head starting has played a pretty prominent role in certain areas in releasing this pathogen into the wild. And so in California, we've done a few surveys in a small number of individuals over the course of the last several years, and not found Amidomyces in the state. But so this project, the field teams were collecting swabs based on our sampling protocol from these animals, and in year one, Tom, let's go to the next slide.
Go to that one.
Yeah, let's go to that one. In year one, it was a combined oral cloacal shell swabs of all Western pond turtles, and we were just focusing on Amidomyces. And in 2024, we did those combined swabs, and we looked for Amidomyces in Western pond turtles, but then we've been doing multi-pathogen surveillance in Western pond turtles and a subset of sliders. Let's go back 2 slides to that table. And the reason we wanted to do that is because across North America, we've been dealing with pathogens that are playing a role in conservation. Some have no effect on the host in which they evolved in. Things like a host-adapted herpes virus like terrapin herpes virus 1 in eastern box turtles, it's supposed to be there. It doesn't really cause a whole lot of disease, but when it gets outside the host, that's when the disease can occur. So in confiscation events, head starting events, or translocation events, the host-adapted herpes that are in the locations and in the environments are incredibly important to understand what the potential risks are to other species. Next slide, Tom. And so we've whittled it down to these turtle pathogens with various different parameters. So high threat to turtle conservation is ranavirus and Amidomyces. They work very differently. Ranavirus is a rapid, almost nearly fatal infection, viral infection, happens within 3 to 4 weeks, can cause 90% mortality in sensitive species. Amidomyces is also a fatal process over time, but this can take multiple years and possibly even up to a decade to cause disease and lesions. And then we've got host adapted ones, and every species has their own. So box turtles have 3 actual herpes viruses, Blanding's turtles have a couple, sliders and spotted turtles have a herpes virus, some adenoviruses. And then we've even seen cancer-associated viruses in populations in the eastern United States. And then there's obviously zoonotic concerns. And so our multi-pathogen surveillance is designed to get a better picture, a whole holistic picture of what we can be detecting. And I think it's important when we're talking about these things that we, kind of differentiate detection from infection from disease. So the qPCRs that we're using are detecting segments of DNA or genetic material that are on these. Does not say that the infectious or the DNA is from infectious organisms at all, or even that they're alive. Whereas infection is that pathogen, a live pathogen, got into the animal, but it may not be causing anything. So those are like the host adapted herpes viruses. They're causing an infection, but it's not leading to disease. Disease is when the host responds with clinical signs meant to fight those infections, and those have both clinical and subclinical impacts. So this surveillance that we did in this project, it was not really detecting for disease. It was detecting pathogen DNA, and we were using that as a correlate for infection and disease, depending on clinical signs that were associated. All right, Tom, let's go to those results. So easy peasy here. 2023, all the Western pond turtles, 259 samples were all negative for Amidomyces. 95% confidence interval of the true prevalence is between 0 and 1.4%. In 2024, we've only tested 134 animals, and the confidence interval is 0 to 2.7%. And the multi-pathogen surveillance is pending. But having said that, since putting these slides together last week, we've gotten some initial results. Some of the red-eared sliders that we tested at the site have herpes virus that we're seeing in sliders in eastern North America. And we have no idea what the impact could be on Western pond turtles at this time. Is there another slide, Tom?
This is Rob's.
Yeah. All right. So overall, I would say that the good news is that Amidomyces is not there. And this is the results that we're seeing in Illinois as well. Like, we have very low prevalence in Illinois outside of head starting parameters. And so this does not mean that Amidomyces is not going to be a problem. In fact, this means that these animals may be susceptible to introductions of Amidomyces. And so translocation events have a much higher risk. They have to be paired with pathogen investigations before and after release of turtles, either through head starting or through translocation events. Animals shouldn't be moving, and non-native species really pose a threat. Red-eared sliders in particular are one of the species that we're detecting this with in Illinois and in the eastern United States. And so, not that there aren't numerous problems with invasive species everywhere, but they're carrying diseases that are likely going to be threatening the conservation efforts of the Western pond turtle and other endangered species. All right, that's it for me.
Thanks, Matt.
Yep.
Thanks, Matt, and thanks everybody on the team. Just wrapping up here. This is a project that's an output of Chris and I in DoD Park and soliciting support and funding, and we're very grateful to Legacy Resource Management Program for funding this to stay on top of our mission-sensitive species. And we do have a defined list and a report that laid out what those species are, and the pond turtles, plural, what started as a single species with that concept is certainly one of those, and it's great to get all this information across such a vast area. I wish it was bigger. I wish we still had them in the Northwest. I will say that we're actively pursuing funding and support to do some eDNA surveys on military properties in the Northwest just to see if we detected them there, where we believe that they're gone, and they are by and large around those installations in those parts of Washington State. A couple of them are relatively close to historical, but that's future directions. But with this study, the establishment of long-term monitoring and a framework within DoD, and to stay on the same page, what you saw was just an abundance of wonderful analyses and outputs from basically 2 years of sampling and the disease and pathogen surveillance. But if we could string together more years, we'll start to resolve some of these patterns and maybe shrink some of those error bars on what was laid out so elegantly by Matt and Emily. So going forward, it's certainly a species of interest for the resource managers on the call and the military personnel. The strategic partnership aspect is phenomenal. We got to cover a lot of ground and interact with a lot of our agencies, NGOs, folks that came out that were just interested in turtles, the public at large, some of the top brass, our officers and military personnel. So this was a great way to do that. Back up, Tom, sorry. And then creating that database. I joke and say if pond turtles were a business is booming on military lands. And we got a lot of data when we compiled what we had for, in some cases, over 20 and almost 30 years worth of information where we had detections of pond turtles on these properties that were surveyed. And so carrying that forward, where we now have that database, the adaptive management, we don't know what the future holds. We're certainly in a biodiversity crisis globally, climate change, and what's been predicted with the species status assessment certainly begs questions going forward and what else will change. But it's nice to have our finger on the pulse now, and we plan to do that going forward. And then the commitment to conservation to make sure, like Tom said, to balance our land uses with species protections. This is a great example of how we're aligning the land uses on our military lands for this particular species. Next, Tom. And I must say this is kind of a short list. It just looks like it. There were a lot of people that we have to thank, and it wouldn't be possible without the support that we got from the installations to give us access and support what we were doing on the ground. So we're ever grateful, and the agencies who permitted us and all the folks who came out to help us. This was a big collaboration for a large-scale project, and we're not done yet. We've still got some work to do. In the next year, we should probably have the final report, and that'll be available to everybody, including Western Pond Turtle RCC. I hope to share the outputs with them, and just wanted to thank everybody for showing up today. We had a good turnout of about 74 people at the top, and that's a good showing for this webinar. So thanks to everybody involved, and if we come knocking for information or collaboration with the Pond Turtle, you heard it here first. Thank you. And with that, we'll take questions. And Daniel, I don't know if you're the shepherd on the questions or if we just want to wing it, but whatever questions may be, they'll probably fall on different folks.
Sounds good. Thank you all of our presenters for sharing your insights and experiences today. We will now begin the Q&A portion. As Rob announced, participants may use the raise hand function and wait to be called before unmuting and asking their question, or participants may submit their questions to the chat. It looks like the first question we have is from Sherry. Sherry asks, how can we access the database or receive data collected on our installations?
Send me an email, but we don't have a final list, but we're happy to provide, like we've done for people with their in-rump stakeholder meetings. Hey, what did you get this year? What did you get this year and last year? We're happy to pull that data out and supply it to you. If you wanted to send me an email, I'd be happy to compile that and send it to any or all installations that ping us. We have given that to some, but when it's a final product, then we will be letting everybody know that the final report is available, and that will have all the information in it.
We have another question from Jennifer. Jennifer asks, for Emily and Matt, curious about the declining capture rate over trapping period. Did you replace bait and traps set for multiple nights or keep the same bait for the duration of the trapping period?
Emily, either of us can answer this.
You go ahead, I can add if I think of something else.
Okay, so our first year, so we had 4 trap nights, and we would set traps on Monday, and then we would rebait on Wednesday, and then pull on Friday. So that's for the first year. The second year, so we only had 2 trap nights. So Emily explained that we would go Monday to Wednesday duration. So we would rebait on that Tuesday as well. So every trap would be same duration. Every trap was rebaited at the same time as well. So there was no just random rebaiting or anything like that, because we wanted to keep it as consistent as possible.
Bait type was also kept consistent, so it was just can of sardines.
Yeah, sardines and oil is what we used for both years.
Carly asks, how successful were the minnow traps versus the standard hoop traps?
So I looked at this last year in 2023 with our data, and there really wasn't a huge difference. Like both traps were catching turtles. I will say the minnow traps, I haven't looked at it. I haven't run analysis on it, but you would find a lot more smaller turtles per se in the minnow traps. Like we would find hatchlings in those that could easily go through the holes in our hoop traps. So other than that, there really isn't like a really noticeable difference. Both are catching turtles. And the minnow trap's another thing. They're easier to kind of, with all the I think we call them Tule out in California, or Tulia. The cattail, it's a lot easier to fit the minnow traps in those versus the hoops, because you have to fight your way to get in those. But again, they both were just as successful.
Andrew asks, is the trapping or monitoring protocol available somewhere?
So that is one of our deliverables for our report and everything. And so, yeah, we will be creating one as well. And then Rob, I saw you perk up, so you can also add on to that as well if you'd like.
Yeah, just we're giving this talk because we're really excited about the results, but it's not quite final yet. So once the final report is done, the final reporting and deliverables are available, we'll have those. But in the interim, if you want some tips on setting up monitoring for this coming year, ping one or any of us, and we can rally the rest of the crew and get you the information that you need for how to go about conducting trapping on your particular lands.
Katrina asks,
But,
how?
Sorry, Daniel.
Sorry.
Tom showed a slide as far as continuing with that answer, the different protocols that have been employed over time, and they're a little dated. I'm not going to say that they're off in any way, and the authors did a fantastic job with those, but it's nice to have the scale of sampling that we did across both species in successive years. And I suspect that we might have some minor changes that we'd recommend compared with those other protocols that are available. And even the author like Rob Fisher on the USGS protocol, if he wrote it again today with his team, then it would probably come out a little bit different. And props to those guys for getting it out. And Bruce Parry on that protocol as well. But Matt, I don't know if you want to offer anything on that, but I don't think it's going to change substantially the outcome. But we do hope to continue growing a better protocol, just like with any sort of scientific endeavor, based on those earlier ones. Did I frost everybody's questions with that?
I'll take that. So we have another question from Katrina. Katrina asks, how did you determine which water bodies to trap on each installation?
What do you want to
So I can cover it if nobody else wants to. Tom mentioned earlier in the presentation, we did meet with all the installation staff that were in charge of wildlife operations, asking for water bodies where known detection was at. So they were the ones that directed us to the water bodies where we had the best chance of catching the highest capture. That was in 2023. In 2024, we revisited that water body in the first half of the week. The second half of the week, we just asked for any other water body where we might have likely detection to help boost Matt's detection model.
And I'll just add, we didn't randomly select across all available habitats. We didn't random stratify. We went with where do we have access and or where are pond turtles, and each installation has a different military mission for the most part, and so our where we are able to access on the base has something to do with that. And then in some cases, like Point Mugu, you really only had one water body that was available for trapping, versus most of the other areas had a lot of water in some cases, and there's no way you could sample it all. So we had to do the best we could. But each location to trap is unique, and I think anybody who's done trapping for pond turtles would say the same. You have to evaluate it based on that particular site and just do the best you can.
Dan asks, have there been any efforts to create or enhance pond wetland habitat for enhancement or reintroduction? And he also asks if anyone has used eDNA for determining occupancy.
Yeah, I see my brothers in the room, and they just finished eDNA, and I'm not sure if anybody else from Edwards Air Force Base, but they just did a big eDNA project out there, and they got hits for western pond turtle in the wetlands where they were trapping them historically and where they were not. And so it has been done. It's been done in other, I can't remember if it might have been a different species I'm thinking of in the Northwest, but not so much pond turtles, but or maybe it has. Anyway, eDNA is out there, and the assays are available, and it can be done. And I'm sorry, what was the other part to the question?
Has anyone used eDNA for determining occupancy?
Yeah, and that's what I just answered. Wasn't there a front end?
Have there been any efforts to create or enhance pond wetland habitat for enhancement or reintroduction?
Yeah, I'd have to leave it to the installations to answer, but I do know that in some cases there have been enhancements made to wetlands, not specific to pond turtles. I don't think it was for other endangered species like steelhead or royal toad or whatever other aquatic species. With I think 90% of all at-risk species in California are generally found in riparian areas, and so there is a lot of that going on. And there's a secondary benefit probably to pond turtles, but I don't think that that's well cataloged.
And the final question I see in the chat is from Tiffany. Tiffany asks if you collaborated with the Fish and Wildlife Service on trapping and monitoring protocol.
They don't have a monitoring protocol. USGS wrote the most recent one. And so we did collaborate with them and we talked to them. We got permits from the state of California. And so we have open dialogue with U.S. Fish and Wildlife Service all the time in all aspects. They're my colleagues. And so I'm happy to have a lot of them on this call today as well. We all are aware of what options are available and how to go about the trapping, I guess, for these species. And we stay in pretty good communication on that.
We just got one final question popped up. Kathleen asked if there's an eDNA protocol, and do you know what the detection probability of the eDNA project at Edwards is?
I'd have to look at the data. I don't remember for Edwards specifically. I do know that it can be hard. For some reason, turtles don't shed as much DNA as some other organisms, and you can miss them with eDNA studies. As far as a protocol, I could get you that, what was employed at Edwards, and I could ask the base, or I know the folks out of Utah State that did the work. And so, Mike Dreslik, do you have any other, or Matt, as far as protocols for eDNA? It's basically whatever you're doing for amidid turtles.
Yeah, it's going to be very similar to amidids, general protocols. Tom might even be able to answer that, because I imagine protocols would be very similar to what they're doing out east for spotteds and wood.
Yeah, I would add that
all the protocols, Tom, are very heavy on how you go about collecting the water. But as long as you're in turtle habitat, I don't think there's a lot of heavy additional legwork, if you will, in the protocols as far as finding where to collect the water.
Yeah, I mean, it would be good to hear from the folks who actually conducted the work on Western pond turtle. Obviously, just like with detection in general, that is what Matt Parry was reporting on. Detection of eDNA is very dependent upon the abundance and density of the individuals and the time of year and the other environmental correlates. So, how you set up protocols for capturing eDNA is very much dependent on the system you're working on and when you do it. And we vary with turtles in the eastern United States, so not in these kind of semi-xeric systems. We've varied those protocols based on the system, whether it's Lovich or lentic or time of year. So that's a pretty empty answer, but it needs specifics from the people who've actually done it.
And looks like that answers all the questions we have in the chat. And presenters, do any of you have a last word you'd like to get in?
I'm just really grateful to have everybody involved in the funding from Legacy. It's a team effort, and many of you that came to this talk were involved in some capacity, and just really grateful for the support and how this is turning out. So thank you everybody for attending and for your support.
Yeah, thanks for support of this project. Thanks for support of the collaboration. Thanks for support of the graduate students. Thanks for support of the species conservation. Fantastic all around.
Yep. We'll see you out there.
And thank you to our wonderful team of presenters for sharing your insights and experiences today. This concludes today's presentation. A recording of the session will be posted on the Natural Resources DNX website under Resources. Thank you everyone for joining us, and have a great rest of your day.
Take care.